We're getting right into it, cutting straight to the physical, chemical, and electrical frontier of life.
The cell membrane.
The cell membrane and the signaling networks it governs. It sounds basic, but it's anything but.
Oh, absolutely. It's an essential deep dive. We often talk about cells like they're just, simple building blocks.
Right.
But the moment you actually look at that boundary, the plasma membrane, you realize it's not just some passive shell. Not at all.
It's more active.
It's the absolute foundation of what makes a cell a cell. It's this highly selective, incredibly dynamic barrier, and it dictates pretty much everything.
Right, what?
Well, how a cell communicates, its electrical excitability, and fundamentally how it maintains a homeostasis, stability, against a pretty chaotic external world.
So without this specific kind of barrier.
Life as we know it, impossible. It's that critical interface.
Okay, let's unpack this then. We are diving deep into the core mechanisms that really define what it means to be alive, starting right at the edge of the cell and then moving into those complex, amplified conversations happening inside. Right, so to understand this cellular frontier, we've got to start with the structure, and specifically, the big idea that really changed cell biology back in the 70s.
Ah, the fluid mosaic model.
Attributed to Singer and Nicholson, right. Before that, people kind of pictured the membrane differently.
They did. It was often seen as more static, maybe like a fixed laminated sheet, a bit rigid.
Which didn't really explain things well.
No, that older view was frankly completely insufficient. It couldn't explain how dynamic the membrane actually is. The term fluid mosaic itself is brilliant
Why is that?
because it captures two key properties at once. It's a mosaic because it's packed with all sorts of different components, proteins, lipids, carbohydrates arranged like, tiles in a complex pattern.
Okay, the mosaic part makes sense. Lots of pieces.
But crucially, it's fluid. These components aren't locked rigidly in place. The phospholipids themselves, and even many of the proteins, are constantly shifting, moving laterally within the membrane.
Like jostling around.
Exactly. And that fluidity is essential for processes like endocytosis, when the cell engulfs something, or for receptors clustering together to start a signal. It has to be dynamic.
And the molecule that really drives the whole structure, the reason it forms in the first place, is the phospholipid.
That's the one.
Yeah.
If you want to grasp why life automatically organizes itself into these sealed compartments in water, you absolutely have to understand the phospholipid. It has this ingenious dual nature.
Amphipathic, is that the term?
Amphipathic, yes.
Okay.
It means it has two personalities.
Yeah.
Chemically speaking. It's got a polar hydrophilic head, loves water.
Okay.
And then two non-polar hydrophobic fatty acid tails, they hate water.
Right. Oil and water don't mix.
Precisely. So put these molecules in any watery environment, inside the cell, outside the cell, and they spontaneously arrange themselves into a bilayer. It's automatic.
How?
The hydrophobic tails desperately try to avoid the water, so they tuck inward, facing each other. The hydrophilic heads, loving the water, face outward towards the aqueous solutions on both sides.
Creating the two layers.
Exactly. It's this self-sealing, energetically favorable structure. That's why membranes are both robust, they hold together, and flexible. It's just fundamental chemistry dictating the geometry of life.
Okay, if it were just phospholipids, wouldn't it be really susceptible to temperature, like melt in the heat or freeze solid in the cold?
That's a great point. Pure phospholipid bilayers are very temperature sensitive.
Yeah.
They would be too fluid, too leaky when warm, and too rigid, almost crystalline when cold.
So how does the cell deal with that? We don't just fall apart when the temperature changes.
That's where cholesterol comes in.
Yeah.
It's often got a bad rap in, dietary discussions. But in the membrane, cholesterol is absolutely essential. It's the membrane's molecular thermostat, its fluidity buffer.
How does it work?
It inserts itself right in between the phospholipid tails. At warmer temperatures, its rigid steroid ring structure kind of restricts excessive movement of the tails. It keeps the membrane from becoming too fluid.
Okay, it stiffens it up a bit when it's hot.
Right. But conversely, at low temperatures, it does the opposite. It disrupts the tight packing of the fatty acid tails, preventing them from solidifying.
So it keeps it fluid when it's cold.
Exactly. It maintains this optimal functional state of semi-fluidity across a range of temperatures. Really quite clever.
So we have this lipid C, buffered by cholesterol. Now we need the functional parts, the machinery. That's the proteins, right? They make up a huge chunk of the membrane mass.
About half the mass, typically, yes.
Yeah.
And we generally categorize them based on how tightly they're associated with the membrane. First, you have the integral proteins. These are permanently embedded, often spanning the entire bilayer. To do that, They need segments, usually alpha helices, made of hydrophobic amino acids that are comfortable sitting within that oily lipid core.
And these are the main workers.
These are your major functional elements, yes. They form the specific channels for ions, the carriers for molecules like glucose, the pumps, and the receptors that detect signals from outside. They bridge the gap.
Okay, integral means embedded. What's the other type?
Then you have the peripheral proteins. These are more loosely associated. They don't dive into the lipid core. Instead, they bind to the surfaces, either to the integral proteins or to the polar heads of the phospholipids.
And what do they do?
They play crucial roles too. Things like anchoring the membrane to the internal cytoskeleton, which gives the cell its shape and structure. They're also really important in the initial steps of intracellular signaling pathways, relaying messages from the integral receptors deeper into the cell.
Before we jump into transport, you mentioned one more structural bit that's maybe overlooked but has huge real-world relevance. The carbohydrates, the identity tags.
Ah, yes, the glycocalyx. This is fascinating and directly connects to things everyone understands, like blood type.
How so?
On the outer surface of the cell membrane, you have carbohydrates attached either to proteins making glycoproteins or attached to lipids making glycolipids. This sugary coat is called the glycocalyx.
And its job.
Its main purpose is cell recognition. It acts like a unique cellular signature, allowing cells to recognize each other, distinguish self from non-self, crucial for the immune system.
And the blood type connection.
That's the classic example. Your A, B, AB, or O blood type is determined entirely by the specific type of short carbohydrate chains attached to glycolipids on the surface of your red blood cells.
Wow, so it's literally a molecular ID badge.
Exactly. And that's why getting the wrong blood type in a transfusion is so dangerous. Your immune system recognizes those foreign carbohydrate tags and attacks. It's cell biology with immediate medical consequences.
Okay, so we have this dynamic, self-assembling, temperature-stabilized, protein-studded wall with ID tags. That's the barrier. Now, the fundamental question, how do things actually get across it, right? The whole point of this complex structure is that it's selectively permeable. Some things pass, others don't. So moving from what it's made of to how molecules actually navigate it. And the first big split is energy, right? Passive versus active.
Exactly. Passive transport is basically things moving downhill, energetically speaking. It follows the second law of thermodynamics. Molecules move from where they're more concentrated to where they're less concentrated, down their concentration gradient.
And it doesn't cost the cell any energy.
Correct. It's free energetically. The simplest form is simple diffusion.
Which is only for a select few molecules.
Very select. Mostly small, non-polar things. Gases like oxygen and carbon dioxide slip right through the lipid bilayer because they can dissolve in that hydrophobic core. Small fatty molecules, too.
But what about most things the cell needs? Water, ions, sugar?
Yeah. The vast majority of important molecules, ions like sodium or potassium, sugars like glucose, amino acids, even water, to some extent they're either charged or polar. That hydrophobic lipid core is like a brick wall to them.
So they need help.
They need help. That's facilitated diffusion. Still passive, still moving down the gradient, but it requires a protein helper.
And the helpers come in two main types, channels and carriers.
That's right. Channels are basically protein-lined tunnels or pores through the membrane. Think of the famous aquaporins specific channels just for water, or ion channels that are specific for sodium, potassium, calcium, etc.
And these are fast.
Incredibly fast. When open, millions of ions or water molecules can whisk through per second. They don't usually bind the substance strongly, they just provide a pathway.
Okay, and carriers?
Carriers, like the GLUT transporters for glucose, are different. They work more like a revolving door. They have to physically bind to the specific molecule on one side of the membrane.
Then what?
That binding triggers a conformational change. The protein literally changes shape, flipping the binding site to face the other side of the membrane where it releases the molecule.
Which sounds much slower.
It is significantly slower than channel transport because of that binding and shape change cycle. And it's saturable. If all the carriers are busy, transport rate maxes out.
Got it. And you mentioned aquaporins for water. Is osmosis, the movement of water, considered facilitated diffusion then?
Osmosis is the net movement of water across a semi-permeable membrane, driven by differences in solute concentration. While some water can sneak through the lipid bilayer directly, the bulk of rapid water movement in many cells happens through aquaporins. So yes, osmosis is often facilitated diffusion of water.
Okay, that covers going with the flow, down the gradient. But life isn't always easy. Sometimes you need to move things uphill.
Absolutely. Often, cells need to concentrate substances against their gradient or push ions out even when they want to come in. That's active transport.
And this costs energy.
This always requires an energy input, usually from ATP hydrolysis. This is where the cell really exerts control.
And the main type uses ATP directly.
Yes, that's primary active transport. The energy from breaking ATP's phosphate bond is directly used to power the movement of ions or molecules against their gradient.
And the absolute superstar example here is...
The NACO-IPASE,
Yeah.
the sodium-potassium pump. It's fundamental to almost all animal cells. It's like the cell's engine.
I remember hearing it uses a massive amount of a neuron's energy budget, just constantly running.
It's tireless. In a resting neuron, it can account for something like a third of all the ATP consumed. It has to constantly work to maintain the crucial gradients of sodium and potassium across the membrane.
Can you walk us through that cycle? It's 3 sodium out, 2 potassium in, right? Why that specific ratio?
That ratio is critical. Here's how it works, roughly. Inside the cell, the pump has high-affinity binding sites for 3 sodium ions. They bind.
Okay.
That binding triggers the pump to phosphorylate itself using ATP. It basically sticks a phosphate group onto itself.
And that changes its shape.
Precisely. The phosphorylation causes a major conformational change. The pump flips open to the outside of the cell, and its affinity for sodium drops, so the 3 NAO ions are released out there.
Okay, sodium's out. What next?
Now, in this outward-facing shape, the pump exposes high-affinity binding sites for 2 potassium ions from the outside. They bind.
Right.
Potassium binding triggers the dephosphorylation of the pump. The phosphate group comes off. This causes it to flip back to its original inward-facing conformation.
Releasing the potassium inside.
Exactly. Its affinity for potassium drops, the 2 ions are released inside, and the pump is ready to bind sodium again. The cycle repeats.
And the 3 NIO out for 2 in. What's the significance?
It's electrogenic. For every cycle, the cell loses 3 positive charges and gains only 2. That's a net loss of 1 positive charge from the inside. This constant pumping action directly contributes to making the inside of the cell electrically negative relative to the outside.
That's amazing. So the pump sets up both concentration gradients and an electrical gradient.
Exactly. It establishes that steep concentration gradient for sodium, low inside, high outside, and potassium, high inside, low outside. And it helps establish the negative charge inside.
Which brings us to the cleverness of biology. Once you've spent all that ATP energy setting up, say, that big sodium gradient,
You can use it. The cell leverages the potential energy stored in that gradient. The fact that sodium is desperately trying to get back in to power other transport processes.
that's secondary active transport.
That's it. It doesn't burn ATP directly itself, but it's completely dependent on the gradient created by a primary pump, usually the NACO pump. It's like using water stored behind a dam to turn a turbine.
And there are different types.
2 main flavors. Co-transport or symport is when the driving ion, like NAIU rushing in, drags another molecule along with it in the same direction. The classic example is the NAIU glucose co-transporter found in your gut and kidneys. It uses the inward sodium gradient to pull glucose into cells, even against glucose's concentration gradient.
So sodium going in powers glucose coming in.
All right. The other type is countertransport or antiport. Here, the driving ion, like NAIU rushing in, powers the movement of another ion or molecule in the opposite direction. A key example is the NACO exchanger.
What does that do?
It uses the energy of NAIU entry to pump calcium out of the cell. This is crucial for keeping the intracellular calcium concentration incredibly low, which is essential for proper signaling.
Okay. Primary uses ATP, secondary uses a pre-existing gradient. What about moving really big things or large amounts?
Ah, right. For that, we need bulk transport. This involves wrapping chunks of material in membrane vesicles. Exocytosis is the process for secretion. Think of a neuron releasing a flood of neurotransmitter molecules. Vesicles filled with the transmitter fuse with the plasma membrane, dumping their contents outside.
And the reverse is endocytosis, taking things in.
Exactly. But let's focus back on those ion gradients set up by the pumps and the resulting electrical charge. This is the membrane potential.
The VM, you hear that term a lot with neurons, usually around minus 70 millivolts.
For a typical neuron at rest, yes, the resting membrane potential, VM, is about negative 70 millivate. The inside is negative relative to the outside.
And that specific number, negative 70 millivolts, isn't arbitrary, right? It comes from the balance of ions wanting to cross the membrane.
It's absolutely not arbitrary. It's a carefully calculated balance. At rest, the membrane isn't equally permeable to all ions. It's much more permeable to potassium, Co, than to sodium, NaOH, mainly because of co-leak channels that are always open.
Okay, so potassium is leaking out.
Potassium is constantly leaking out down its concentration gradient. As positive co-ions leave, the inside becomes more negative. This negative charge starts to pull Co back in. The equilibrium potential for an ion is the voltage at which the electrical pull exactly balances the concentration gradient push, so there's no net movement.
And the resting potential is sort of an average of these, weighted by permeability.
That's a great way to put it. The Goldman-Hodgkin-Katz equation describes it formally, but conceptually, Vm is a permeability-weighted average of the equilibrium potentials of the main ions. Since co-permeability is highest at rest, Vm, Nika 70 milliV, is closest to the co-equilibrium potential.
So what are those equilibrium potentials? Let's get specific, say, for a mammalian neuron around body temp.
Okay, these numbers really set the stage for understanding excitability.
Yeah, this feels important.
Potassium. Its equilibrium potential, EK, is around negative 90 milliV. Since the resting potential is only 70 milliV, Co still has a net driving force pushing it out of the cell, making the inside more negative. That outward leak is key to the resting state.
Okay, Co wants out, driving towards negative 90. What about sodium?
Sodium, NEO. Its equilibrium potential, ENA, is way up at about plus 60 milliV. Now, compare that to the resting Vm of negative 70 milliV. There's a huge difference. Sodium is being pushed in by both its steep concentration gradient, high outside, low inside, and the strong electrical attraction, positive NAU pulled towards the negative inside. The electrochemical driving force on NAU is enormous, wanting to rush in.
Which is why opening sodium channels causes such dramatic change, like in an action potential.
Exactly. That influx drives the membrane potential rapidly towards plus 60 millimeter. Now let's look at calcium.
Even more extreme, I gather.
Even more extreme. Ekeldo is around plus 120 millimeter. The concentration of free Cu inside the cell is kept incredibly low, like 10,000 times lower than outside. So the driving force for Cu entry is astronomical.
Wow.
When a calcium channel opens, the resulting influx is like opening a fire hose. It's a very powerful, often localized signal used for critical, rapid events, muscle contraction, neurotransmitter release.
And the last main one, chloride.
Chloride. Its equilibrium potential, ECL, is often quite close to the resting membrane potential, maybe around negative 70 milliV itself in many neurons. This means its net movement depends heavily on small, local shifts in VM or active transport mechanisms. Often, coal influx acts like a brake, making the cell more negative, hyperpolarizing, and harder to excite.
So this resting state, this negative 70 milliV, is this delicate balance. Call leaking out, the NACO pump constantly correcting that leak and pushing NAO out, keeping those massive driving forces for NA and call poised, ready for action.
Perfectly summarized. It's an energy-expensive, dynamic, steady state, primed for receiving and transmitting signals.
Okay, we've got the structure, we've got molecules moving across. Now let's talk about moving information. This feels like the next level up in complexity.
It is. If transport is about moving substances, signaling is about moving messages. This is signal transduction.
Taking an external cue and turning it into an internal action.
Exactly. The basic flow is an external stimulus, like a hormone or neurotransmitter, the ligand, binds to a specific receptor protein on the cell surface. That binding triggers transduction, a cascade of events inside the cell, often involving multiple steps, and crucially, amplification.
Amplification seems key.
It's fundamental. One signal molecule outside can lead to thousands, even millions, of activated molecules inside. This cascade ultimately leads to a cellular response.
And what kind of responses are we talking about?
Almost anything a cell does. It could be activating or inactivating enzymes to change metabolism, switching genes on or off in the nucleus to change protein production, triggering muscle contraction, initiating cell division, or even triggering programmed cell death.
And so what here? I mean, this isn't just abstract biology, is it, when signaling goes wrong?
That's when things really go wrong systemically. Defects in these communication pathways are at the heart of so many major diseases. Cancer is essentially a disease of runaway signaling, uncontrolled growth signals. Diabetes involves major problems with insulin signaling, which controls metabolism. Many neurological and psychiatric disorders involve imbalances in neurotransmitter signaling pathways. It's absolutely central to health and disease.
Before we get into the specific hardware, the receptors, let's quickly map out the different distances of communication, how cells talk to each other.
Good idea. We can categorize signaling based on how far the signal travels. The most intimate is autocrine signaling.
Cell talking to itself.
Pretty much. A cell releases a signal that binds to receptors on its own surface. This often acts as a way to reinforce a decision or a developmental pathway.
Okay, then maybe neighbors.
That's paracrine signaling. Here, a cell releases local mediators, things like growth factors or cytokines, that act only on nearby cells in the immediate environment. It's like local gossip within a tissue.
Makes sense. What about long distance?
That's endocrine signaling. This uses hormones, which are secreted into the bloodstream and travel throughout the body to reach target cells that could be very far away. This is slower, but the effects are often widespread and long-lasting. Think adrenaline or insulin.
And the nervous system has its own special version.
Absolutely. Synaptic signaling is unique to neurons. It's incredibly fast and precise. A neuron releases neurotransmitters across a tiny gap, the synapse, to act directly on the target cell, which could be another neuron or a muscle cell. It's highly localized and very rapid.
Is there one more? Direct contact.
Yes, juxtacrine signaling. This requires direct physical contact between cells. Membrane-bound signals on one cell bind to receptors on an adjacent cell. The Notch signaling pathway, crucial during development for cells to decide their fates based on their neighbors, is a prime example of this contact-dependent communication.
Okay, lots of ways to talk. Now, the hardware. The receptors themselves. Where should we start? Maybe with the biggest target for drugs.
We absolutely have to start with the GPCR's G protein-coupled receptors. These are just a massive family of receptors.
Why are they so important pharmacologically?
Their sheer ubiquity and the variety of critical processes they control: vision, smell, neurotransmission, hormone responses. Structurally, they all share this characteristic feature: a single polypeptide chain that snakes back and forth across the membrane 7 times.
The 7 transmembrane domain structure.
Exactly. And because they're involved in so many things, it's estimated that something like maybe 50% of all modern drugs on the market target GPCRs in some way.
Half. Wow. That includes things like antihistamines, beta blockers.
Antihistamines, opioids, beta blockers for heart conditions, drugs for asthma, psychiatric medications. The list is enormous. Understanding GPCRs is fundamental to pharmacology.
So how does that 7-pass structure actually transmit a signal inside the cell? What happens when the hormone or neurotransmitter binds on the outside?
Okay, so the ligand binds to the extracellular part of the receptor. This causes a subtle but critical change in the receptor's overall shape, particularly on the intracellular side.
Right.
This shape change allows the receptor to interact with a specific partner protein waiting just inside the membrane. A G protein. These G proteins are trimers. They have 3 subunits: alpha, beta, beta,
And gamma.
and they're just hanging out there.
They're associated with the inner leaflet of the membrane, waiting. In its inactive state, the alpha subunit has a molecule called GDP, guanosine diphosphate, bound to it.
Okay.
When the activated receptor bumps into the inactive G protein, it acts like a catalyst. It causes the alpha subunit to release its GDP and immediately bind a molecule of GTP, guanosine triphosphate, instead.
GDD out, GTP in. That's the switch.
That is the critical on switch. Binding GTP causes the alpha subunit to change its shape. This makes it dissociate from the beta gamma dimer.
So the trimer splits.
Grid splits. Now you have 2 active signaling complexes: the alpha subunit with GTP bound and the free beta gamma dimer. Both of these can now move along the membrane and interact with their specific downstream target proteins or effectors.
Effectors like enzymes or ion channels. Exactly. Things like adenylyl cyclase or phospholipase C, or sometimes ion channels directly. So one receptor activation leads to potentially many G proteins being activated, and each of those activates an effector. That's the start of amplification.
And how does the signal get turned off? Does the G protein just stay active forever once GTP is bound?
No, thankfully. The system needs to reset. The alpha subunit has an intrinsic built-in timer. It's a slow GT pace.
Meaning it breaks down the GTP.
Right. It slowly hydrolyzes the bound GTP back to GDP. Once it's GDP bound again, it loses its affinity for the effector, reassociates with the beta gamma dimer, and the whole G protein becomes inactive again, ready for the next signal.
But you said it's slow. How does the cell get fast responses?
Ah, good point. The intrinsic GT pace activity is often too slow for rapid signaling. So cells have other proteins called RGS proteins, regulators of G protein signaling. These act as gene pace accelerating proteins. They bind to the active alpha GTP subunit and dramatically speed up the GTP hydrolysis.
So they help turn the signal off quickly.
Exactly.
Yeah.
They ensure the signal is brief and precisely timed, which is crucial, especially in the nervous system.
Okay, so GPCR activates G protein activates effectors. What are the main pathways these effectors trigger?
2 major pathways dominate GPCR signaling. The first involves the enzyme adenylyl cyclase. When activated by a G protein alpha subunit, often called Gs for stimulatory, adenyl cyclase converts ATP into a small molecule called cyclic AMP, CMMP,
The famous second messenger.
the classic second messenger discovered by Earl Sutherland. HAM-AMP then diffuses through the cytoplasm and activates a key enzyme called protein kinase A, PKA.
And kinase add phosphate groups to things, right?
Right. PKA phosphorylates a whole range of target proteins, other enzymes, ion channels, transcription factors, changing their activity and leading to the cell's ultimate response, which could be anything from breaking down glycogen for energy to increasing heart rate.
Okay, that's the CAMP pathway. What's the other main one?
The other major route involves the enzyme phospholipase C, PLC, often activated by a different type of G protein called GQ. PLC's job is to cleave a specific lipid molecule in the membrane called PIP2
Cleaves it into what?
into two different second messengers, inositol trisphosphate, IPRs, and diacylglycerol, DAG. These two then trigger parallel downstream effects.
So one reaction makes two signals.
Yes. IPS is small and water-soluble, so it diffuses into the cytosol and binds the special receptor channels on the endoplasmic reticulum, ER, which is a major storage site for calcium ions inside the cell.
And that releases calcium.
Causes a rapid release of stored Kr from the ER into the cytosol. Calcium itself then acts as another crucial second messenger. Meanwhile, D stays embedded in the membrane, and along with the released Kr, helps to activate another important kinase called protein kinase C, PKC.
And PKC, like PKA, phosphorylates targets.
Exactly. PKC phosphorylates its own set of target proteins, often involved in things like cell growth, differentiation, and managing ion transport. So the PLC pathway activates both Kr signaling and PKC signaling.
Wow, okay. GPCRs are clearly huge. What's the next major class of receptors?
The next big group, especially important for growth, survival, and metabolism, are the RTKs, or receptor tyrosine kinases.
How are they different from GPCRs?
Structurally, they're usually single-pass transmembrane proteins, just one stretch crossing the membrane. And their activation mechanism is totally different. It relies on dimerization.
Dimerization, meaning they pair up.
Exactly. Usually these receptors exist as inactive monomers. When the specific signaling molecule, often a growth factor like epidermal growth factor EGF or insulin binds, it causes two receptor monomers to come together and form a dimer.
Okay, they bind the signal and they pair up. What does pairing up do?
The pairing brings their intracellular domains close together. And these intracellular domains are kinases. Specifically, they phosphorylate tyrosine amino acids. The key is they activate each other through autophosphorylation. Once dimerized, the kinase domain of one receptor monomer phosphorylates specific tyrosine residues on the tail of the other receptor monomer, and vice versa. They cross-phosphorylate each other.
So they basically tag each other with phosphate groups on tyrosines.
Precisely. And these newly phosphorylated tyrosines act like docking sites or landing pads for a whole suite of intracellular signaling proteins.
Ah, so the phosphates recruit the next players in the cascade.
Exactly. Adapter proteins, like one called GEMDO, recognize and bind to these phosphotyrosines. GERB2 then recruits another protein, like SOS, which is a guanine nucleotide exchange factor, GEF, for a small G protein called RAS.
RAS. I've heard that name in relation to cancer.
Yes, RAS is hugely important and often mutated in cancer. SOS activates RAS by helping it swap GDP for GTP, similar to how GPCRs activate the larger G proteins. Active RAS GTP then triggers downstream signaling cascades.
Which cascade?
Two of the most critical pathways downstream of RTKs and RAS are: 1. The MAPK cascade, mitogen activated protein kinase. This is a chain of kinases that ultimately leads to changes in gene expression and protein activity controlling cell proliferation, differentiation, and survival. It's the major growth-promoting pathway.
Okay, MAPK for growth. What's the other?
The other major one is the PI3K right arrow ACT pathway. PI3 kinase is another enzyme recruited to the activated RTK complex. It generates lipid second messengers in the membrane, which then activate the kinase ACT, also known as protein kinase B. ACT is a master regulator of cell survival. It promotes metabolism and actively inhibits apoptosis, or programmed cell death.
So RTKs trigger growth and survival signals.
That's why they're so critical, and also why their malfunction is so central to cancer.
Makes sense. If those signals get stuck on.
You get uncontrolled proliferation and cells that refuse to die, hallmarks of cancer. Mutations that cause RTKs like HER2 in breast cancer or EGFR in lung cancer to dimerize and activate even without a growth factor signal, or mutations in downstream players like RAS that lock them in the active state, are incredibly common drivers of tumors. Understanding this pathway is key to developing targeted cancer therapies.
Right. Okay, GPCRs, RTKs. What's the third class of receptor?
The third major group are the ion channel coupled receptors, also called ligand-gated ion channels. We touched on these briefly with transport, but here the channel is the receptor.
So the signal molecule directly opens or closes a channel.
Exactly. Binding of the ligand, typically a neurotransmitter, causes an immediate conformational change that opens the channel pore, allowing specific ions to flow across the membrane, changing the membrane potential very rapidly.
Example.
The classic example is the nicotinic acetylcholine receptor found at the neuromuscular junction. When acetylcholine binds, the channel opens, letting no rush into the muscle cell, causing depolarization and triggering contraction. It's incredibly fast signal transmission.
And these are targets for drugs, too.
Oh, yes. Drugs that block or modulate these channels are used as anesthetics, muscle relaxants, anti-anxiety drugs, and treatments for epilepsy. Plus, you have the voltage-gated ion channels, like NO, CO, CO channels involved in action potentials, which, while not ligand-gated receptors in the same sense, are crucial communication hardware targeted by many drugs like antiarrhythmics and some anti-epileptics.
Okay, one more category. What about signals that can get inside the cell?
Right. The final class relies on the fact that some signaling molecules are small and hydrophobic, or lipophilic, meaning they can dissolve in lipids.
So they don't need a surface receptor.
They can simply diffuse right across the plasma membrane and even the nuclear membrane. These are signals like steroid hormones, cortisol, estrogen, testosterone, thyroid hormone, vitamin D, retinoids.
And where do they find their receptors?
The receptors, the intracellular receptors, are waiting for them inside the cell, either in the cytoplasm or directly within the nucleus.
And what happens when the signal binds the internal receptor? It doesn't start a cascade like the others.
Generally, no. The activated hormone receptor complex itself undergoes a conformational change that typically allows it to move into the nucleus if it wasn't already there, and bind directly to specific DNA sequences.
DNA sequences.
Yes, called hormone response elements, HREs, located near the genes that the hormone regulates. Binding of the hormone receptor complex to the HRE usually acts as a transcription factor, either activating or repressing the transcription of those target genes into mRNA.
So it directly changes which proteins the cell is making.
Exactly. It leads to a slower, but often much more profound and long-lasting change in the cell's function and characteristics by altering its protein profile. Think about the effects of puberty hormones or the stress response mediated by cortisol. Those involve widespread changes in gene expression regulated by these intracellular receptors.
Okay, we've covered the main receptor types. You keep mentioning second messengers like CAM-MP and calcium. Let's focus on them and that idea of amplification.
Absolutely critical. Amplification is maybe the most striking feature of these cascades. Think about it. One single hormone molecule binding to one GPCR on the outside might lead to the activation of, say, 10 G proteins.
Okay.
Each of those G proteins might activate one adenyl cyclase enzyme, but that enzyme, once active, can churn out hundreds or thousands of CAM-MP molecules.
Ah, there's the first big amplification step.
Right. And then each CAM-P molecule activates a pKa molecule. And each pKa molecule can then phosphorylate hundreds or thousands of downstream target proteins.
So you get this exponential increase in the signal strength.
Exactly. A tiny initial stimulus generates a massive, swift intracellular response. Second messengers are the key agents in these amplification steps. We mentioned the main ones.
Activate PK.
Activates pKa. Its levels are tightly controlled not just by adenyl cyclase making it, but also by enzymes called phosphodieesterases, PDEs, that constantly break it down, ensuring the signal is transient.
Then IPA and DAG from the PLC pathway.
IP triggers CATA release from the ER. DAG activates PKC at the membrane.
And calcium itself. You called it universal.
Calcium ions are probably the most widespread and versatile intracellular signal. That rapid spike in cytosolic CAT, triggered by IPO or by influx through channels, mediates an incredible range of fast cellular responses. Muscle contraction, secretion of hormones and neurotransmitters, fertilization, changes in cell motility, gene expression, it's everywhere.
How does the cell interpret such a simple signal, just an increase in calcium ions, to do so many different things?
That's where calcium binding proteins come in. The most famous is calmodulin. It's a small protein that's abundant in the cytosol. When intracellular cal-Will levels rise, calmodulin binds up to 4 calcium ions.
And that binding changes it.
It causes a dramatic conformational change in calmodulin. This cal-calmodulin complex can then bind to and activate a whole host of other downstream proteins, including important kinases like CAM kinases and phosphatases. So calmodulin acts as a sensor, translating the simple cal-Will spike into specific downstream effects depending on which CAM binding proteins are present in that cell type.
Okay, so calmodulin helps decode the calcium signal.
It's a key player in decoding that universal signal. And of course, the cell has powerful pumps working constantly to sequester CalOil back into the ER or pump it out of the cell to bring the concentration back down very quickly after a signal, readying it for the next spike.
Which brings us to the final crucial part of signaling. Turning it off, signal termination, seems just as important as turning it on.
Absolutely vital. If signals weren't terminated efficiently, cells would be overwhelmed, unable to respond to new stimuli, and locked into potentially harmful states like constant contraction or proliferation. Control requires an off switch.
So how does the cell shut things down?
It happens at multiple levels, kind of mirrors the activation steps. First, the initial signal, the ligand, needs to be removed. It might be degraded by enzymes in the extracellular space, like acetylcholine being broken down, or it might be taken back up by the signaling cell or surrounding cells, or it might simply diffuse away.
Okay, get rid of the trigger. What about the receptor?
The receptor itself often gets desensitized. Even if the ligand is still present, the receptor might be temporarily modified, often by phosphorylation by specific kinases, like GRKs for GPCRs, which makes it less able to activate G protein,
Like it gets tired.
sort of. Or these phosphorylated receptors can recruit proteins called arrestins, which physically block the interaction with G proteins and can also target the receptor for removal from the membrane via endocytosis.
So silence the receptor. What else?
The G proteins need to inactivate themselves by hydrolyzing GTP, often sped up by RGS proteins, as we discussed.
Right, the built-in timer.
Then all those second messengers have to be eliminated. CMP is broken down to AMP by PDEs, IP is rapidly dephosphorylated, DAG is metabolized, and calcium is furiously pumped away.
Clear out the internal messengers.
And finally, arguably most importantly, all the proteins that were activated by phosphorylation by kinases like PKA, PKC, or RTKs need to be dephosphorylated. This is the job of protein phosphatases.
The opposite of kinases.
Exactly. They constantly work to remove those phosphate groups, resetting the target proteins back to their inactive state, ready for the next round of signaling. The balance between kinase and phosphatase activity is absolutely critical for controlling the duration and intensity of signaling pathways. So when you step back and look at all these pathways, you start to see that the real power isn't just in one pump or one receptor, it's how they all connect and interact. These interactions create emergent properties that define how the cell behaves as a whole.
We've definitely hit on amplification, small signal, big response, but you also mentioned crosstalk and feedback loops.
Right. Crosstalk is huge. It means signaling pathways aren't isolated highways, they're interconnected networks. The signals generated by a GPCR pathway, like KMP or CARO, can influence the activity of proteins in an RTK pathway and vice versa.
So the cell is integrating information from multiple sources.
Precisely. It allows the cell to make much more nuanced decisions based on the combination of signals it's receiving, rather than just responding to one signal in isolation. It's like having multiple advisors giving input before making a decision.
And feedback loops.
Feedback loops provide control and stability, or sometimes rapid change. Negative feedback is common for homeostasis. Think of high blood glucose triggering insulin release, which then lowers blood glucose, which in turn reduces insulin release. It keeps things in balance.
Keeps it from overshooting.
Exactly. Positive feedback is less common, but important for rapid all or none responses. Oxytocin release during childbirth is a classic example. Contraction triggers more oxytocin, which triggers stronger contractions. Or the rapid opening of voltage-gated sodium channels during an action, potential depolarization, opens channels, which causes more depolarization. It drives a process to completion quickly.
This whole network sounds incredibly intricate and finely balanced, which must mean that when things break.
The consequences can be severe and widespread. Let's start with problems in the basic transport machinery. Channelopathies.
Diseases caused by faulty ion channels.
Exactly. We mentioned cystic fibrosis earlier, caused by mutations in the CFTR chloride channel, leading to thick mucus because ion and water transport is disrupted in lungs, pancreas, etc.
Right.
But there are many others. Certain inherited forms of epilepsy are caused by mutations in voltage-gated NAU, or CAW channels in neurons, leading to hyperexcitability and seizures. Similarly, mutations in cardiac ion channels can cause dangerous arrhythmias, irregular heartbeats, because the timing of electrical signals in the heart is disrupted.
So just a tiny fault in one channel protein can have life-threatening effects.
Absolutely. Now scale that up to signaling pathways controlling growth. That brings us to cancer.
Which you said is fundamentally a disease of signaling.
It is. Uncontrolled cell proliferation and the ability to evade death signals are key hallmarks. And these are almost always driven by mutations that cause signaling pathways, particularly the RT-KRAS-MAPK pathway, to become permanently switched on.
Like the HER2 or EGFR examples?
Exactly. Or mutations in RAS itself, locking it in that active GTP-bound state, constantly telling the cell to grow and divide, even when it shouldn't. Or mutations that disable tumor suppressor genes, which normally act as brakes on these pathways. It's like the accelerator is stuck down and the brakes have failed.
What about metabolic diseases? You mentioned diabetes.
Type 2 diabetes is a prime example of signaling failure at the reception level. The pancreas might still make insulin, but the target cells, particularly muscle and fat cells, become insulin resistant.
Meaning the insulin receptor doesn't respond properly?
Right. The insulin receptor, which is an RTK, or the downstream signaling cascade involving PI3K and ACT, doesn't get activated efficiently upon insulin binding. As a result, the cell fails to take up glucose from the blood, effectively leading to high blood sugar. The communication is broken.
And finally, the brain. It seems like the ultimate signaling machine, so it must be vulnerable.
Extremely vulnerable. Many neurological and psychiatric disorders involve disruptions in neurotransmitter signaling. For instance, schizophrenia has long been linked to complex dysfunctions in dopamine signaling, much of which occurs through GPCRs.
Okay.
The devastating loss of motor control in Parkinson's disease is directly caused by the death of dopamine-producing neurons and the resulting loss of dopamine signaling via GPCRs in a brain region called basal ganglia.
And Alzheimer's.
Alzheimer's disease is incredibly complex, but growing evidence points towards significant collo dysregulation within neurons as a key contributing factor. Problems with calcium handling, possibly linked to faulty channels or signaling pathways, might lead to chronic low-level toxicity or make neurons more vulnerable to damage and death, a process sometimes called excitotoxicity. That potent calcium signal becomes destructive when uncontrolled.
It's striking how these fundamental mechanisms, when they go wrong, underpin such diverse and serious diseases.
It really highlights how essential this intricate communication network is for normal function at every level, from single cells to the whole organism.
But the flip side is that understanding these pathways, down to the molecular details we've discussed, gives us the power to intervene. That's pharmacology.
Precisely. Knowing the players and how they interact allows us to design drugs that can modulate these pathways with increasing specificity.
Like the PDE inhibitors you mentioned.
Exactly. We know CAMP signals are terminated by PDE enzymes. So drugs like sildenafil, Viagra, inhibit specific PDEs, preventing KMP, or actually CGMP in that case, breakdown. This prolongs the signal, leading to smooth muscle relaxation and increased blood flow.
And beta blockers.
Beta blockers are antagonists for specific GPCRs, beta adrenergic receptors, that normally bind adrenaline. By blocking adrenaline's binding site, these drugs prevent the fight or flight signals from overstimulating the heart, thus lowering heart rate and blood pressure.
And for cancer, targeting those faulty RTKs.
That's a huge area. We have kinase inhibitors, small molecules designed to get inside the cell and block the ATP binding site of specific mutated kinases like EGFR or BCR-ABL, preventing them from phosphorylating their targets. We also have monoclonal antibodies, like Herceptin for HER2 positive breast cancer, which are large proteins designed to bind to the outside of the receptor, preventing the growth factor from binding or stopping the receptors from dimerizing. These targeted therapies are often much more effective and less toxic than traditional chemotherapy.
So we've journeyed from really the oily nature of a single lipid molecule creating a barrier.
All the way to the complex signaling breakdowns in cancer and Alzheimer's and the drugs designed to fix them.
It definitely confirms that the cell isn't just a bag of goo. It's this incredibly sophisticated, self-regulating electrical and chemical computer. And it's all built on these fundamental mechanisms of structure, transport, and communication we've unpacked.
And it's amazing to think about the pioneers who figured this stuff out. You have to remember people like Hodgkin and Huxley, who worked out the ionic basis of the action potential using squid giant axons' incredible work.
Yeah.
Earl Sutherland, who gave us the whole concept of the second messenger with CAMP, and more recently, Nobel laureates like Lefkowitz and Kobilka, who finally determined the detailed structure of GPCRs, which was crucial for rational drug design. And figures like Tony Hunter and Joseph Schlesinger, who were pivotal in discovering and understanding RTKs and their role in signaling.
We really do stand on the shoulders of giants. Their insights gave us the blueprints, but the complexity of the actual cellular architecture, especially that crosstalk and the feedback loops, it's still kind of mind-boggling.
It is, and it means that almost any drug we introduce, targeting just one node, is likely to have ripple effects throughout that network, sometimes unexpected ones.
So here's a final thought for you, our listener, to mull over. Consider how one tiny molecular change, maybe just a single amino acid swapped in a receptor protein, or a gene like HER2 getting copied too many times, how that can flip a switch. How it can transform a well-behaved, functional cell into an uncontrolled, proliferating cancer cell.
And reflect on how much power, both for generating life's processes and for causing catastrophic failure, lies in controlling the flow of something as seemingly simple as a calcium ion.
This network is incredibly complex, incredibly powerful. What stands out to you most in this amazing interconnected system?